response to the number of cells that we target with nearly 50% at the highest dose here within cervical and thoracic, and nearly 70% in lumbar region. We're being, I think, conservative on our quantification here of positive motor neurons, but clearly you see a dose response. We've also then, that's a subjective test. Someone has to score, is that cell positive, is it not positive? We've also taken the cervical, thoracic, and lumbar regions, isolated RNA from those regions, and performed digital droplet PCR to measure GFP expression. And I think once again you can see that if you look at a very molecular approach, that you see a dose response in the amount of GFP transcript that is being produced. Okay, that's GFP. Will it hold true for SMN? And this is now utilizing, instead of GFP, we've went back and repeated this with these various doses with virus expressing the SMN transgene. And once again, you can see a dose response with increasing doses, meaning increasing in the amounts of SMN protein. Why are we going to such detailed efforts? Well, it would be very nice to be able to correlate how much SMN do we need in the spinal cord at cervical, thoracic, and lumbar to result in a mouse to live X number of days, and I'll show you the survival curve. But just think about the human clinical trial as we're marching forward, and we hope that this doesn't happen, but what if a patient passes away in the clinical trial? We have put into the clinical trial for a consent to perform an autopsy and to evaluate the spinal cord and to evaluate SMN levels. We will know and be able to go back and see, do we have any expression in the spinal cord, and be able to correlate each level and come back and look at the mouse data to determine where we sit on this correlative line towards survival. So here is the survival. Once again, we have a very robust increase of lifespan, with the highest dose of animals giving a median lifespan of 282 days. With 25% of these animals given ICV delivery of the AAV9 living past 400 days. So once again, quite striking survival results of these animals, with a dose response, again, of increasing survival within these animals. Does this express GFP positive cells in the lumbar region of the spinal cord? Here we actually injected AAV with a contrast agent into the L3 region into the CSF of a non-human primate, and you can see the CSF traversing here with where we're pointing to on these lines. We can see over 50% of cells in cervical, 60% in thoracic, and nearly 80% of motor neurons in the lumbar spinal cord expressing GFP. One of the things that we had to work out was a short-term tilting to increase the amount of virus that was targeting the cervical and thoracic regions of the spinal cord. And this is no tilting. We're utilizing a Trendelenburg tilting table, a 5 to 10 minute increase of tilting at 130 degrees increased the amount of GFP in the upper levels of the spinal cord. And again, we've went and quantified the amount of GFP positive cells at each of the regions of the spinal cord following these tilting, and furthermore measured the levels of GFP that one can see. Again, the tilting had an effect in each of the cases in cervical and thoracic. Lumbar is strikingly always constant. We target motor neurons in the lumbar region of the spinal cord quite efficiently, but there is an improvement in cervical and thoracic in the numbers as well as levels of GFP positive cells. does this get into the brain stem and into the brain? So I think this is something that we sometimes forget because we're always thinking about the lower motor neurons. We need to pay attention to the motor cortex and to the brain stem nuclei that is regulating respiration. So here is looking at the brains. This is a sagittal section. Motor cortex is here. You have to trust me, there is a section on here. This is our negative control, so there's little to no background staining with our control PBS-treated non-human primate. Looking at the hippocampus, motor cortex, over 50% of the motor cortex is clearly transduced with a GFP vector. This is looking at the hypoglossal nucleus of the brainstem, where we're targeting about 60% of the motor neurons. Trigeminous, almost 80% of the CHAD-positive cells are expressing green fluorescent protein. While we love motor neurons in my laboratory, with this picture of the cerebellum and the cerebellar Purkinje cells, we think we might start studying these cells here with the artwork of transduction of the cerebellar Purkinje cells. We are doing the biodistribution, so where does this virus go once we administer it into a non-human primate? The Food and Drug Administration always wants to know where your product goes. And we're in the middle of safety studies in mice and non-human primates. And so far, up to almost 6 months in non-human primates, we've had a very safe and well-tolerated delivery. One of the things that I've worked on with Dr. Arthur Burgess is a larger animal model of spinal muscular atrophy. So the mouse can only get us so far. We only have a 2-week window to really test therapy. What we've not been able to test is, and we get this question for any of the clinicians in the audience, you have to answer these questions. What will happen to my baby? What will happen when they're weak? Can any drug that's increasing SMN have any effect on reversing this disease? And I think this is early results, but hopefully these pigs will allow us to really answer some of those important questions. Arthur's laboratory built a ramp, and the pigs get fed marshmallows, which they love, and that's a normal pig running up a ramp. How we've done this model is we've developed an SH, small hairpin RNA, that reduces SMN, that's specific towards porcine SMN. So it doesn't touch our therapeutic human SMN that the company calls Charisma. So we're knocking down pig SMN, and then we're going back and we can replace with Charisma as our therapy. So this is what a pig looks like. It's treated at day 5 with CSF delivery of the knockdown. And this is beware, we have created SMA in a pig. So this pig clearly has hind limb weakness. It will get forelimb weakness, and we euthanize the pig before it cannot get to its food and water for humane reasons. This pig shows a drop-off of motor units by Muni, as well as a loss of CMAP. I don't have time to show you all of the electrophysiology, but it clearly shows electrophysiological properties of SMA. Now, hopefully you'll be impressed. We tried to stack the experiment to our favor. That is, we knocked down this pig on day 5. So if I get back in at day 6, can I prevent anything from happening? So knockdown at day 5, replaced with Charisma at day 6. And this is looking at the pig at the same time point at day 49. Ultimately, we've prevented that pig from developing SMA, and it runs up the ramp. Now, the next experiment, so that's what we're calling early treatment. That is, if we had newborn screening in place, that's what we think would happen. Now what happens when we knock down, wait until the pig gets weak by stumbling on its hind limbs, and then administer Charisma? Will we reverse the symptoms? Will we stop the symptoms? So that pig couldn't get up the ramp. That pig had gotten weak. It still has hind limb weakness, and we did not improve Muni. You can't improve Muni, I don't think, unless someone tells me. Once you've lost a motor unit, you're likely not going to get that motor unit back. But this animal actually had an increase in CMAP in the hind limbs. So quite encouraging. I don't have all of the data to show you today, but this pig got slightly stronger in its hind limbs, but we stopped the disease in its tracks, as it certainly did not get worse here, and the forelimbs did not progress. So we're excited about this, and I think this pig will allow us to answer a lot of questions, and it's actually showing us in a larger species that our product that has been made for human clinical trials actually works in a larger species. What are all the check boxes? This is just to show lots of check boxes. I know I'm running close to the end here, so I'm going to move past here, but there's an arduous process here. Other than the fact that the trial opened on April 25th of this year. It's a phase 1/2 clinical trial. It's at Nationwide Children's Hospital, led by Dr. Jerry Mendel, our center director. 2 cohorts at least as planned. This might be changing to a 3x3x3 low, medium, high dose with a potential expansion to larger patients, to a larger number of patients for a pivotal clinical trial. We produced our own good manufacturing practice production of this, suitable to go into patients. And actually, our GMP facility made history by making the largest amount of viral product in a GMP facility with just over 2.5 times 10 to the 16th vector genomes. So on May 13th, history was actually made based upon the fact that a 6-month-old type 1 patient, so a human being was dosed with the largest amount of viral particles on May 13th than any other human being. This baby had 400 trillion viral particles that was pumped into her bloodstream, and it was as if Dr. Mendel had not touched her, which was, I could finally go to bed probably 96 hours after the injection, and that was, we were monitoring for any adverse event. There was no adverse event noted for this. This patient was 6 months old on May 13th and just had her 10-month-old birthday and is approaching her 11-month birthday. So we're excited about this. There's 3 patients that have been dosed, 1 being last week. The second patient has been dosed for just over 1 month as of today. The trial is 0 to 9 months of age. All of this information is available on clinicaltrials.gov. Some of the outcome measures, you'll be hearing about this in our next talk, so I will not spend any time talking about our motor functional outputs because we have the expert talking in the next talk here. Other than I do want to test the audience. We have electrophysiology either by the standard approach that is utilizing needles to do CMAP and Muni. A needle is injected to measure, versus what Stuart Rutcove has developed, an electroimpedance myography non-invasive method. Which of these methods do you think makes a child cry more? The needle, right? Well, that's exactly, okay, there's no real statistics here, but we're part of this NeuroNext where they're doing both of these on the patients. The one that makes the babies cry more is the non-invasive method, and that is, it's a piece of metal that's slightly colder, and I think we think it's scaring the babies, and the babies will cry when the non-invasive is put next to their skin, and they do just fine here with the needles. Lots of clinical trials. These patients will be followed over 2 years. I think things that we've been thinking about, not only within our research laboratories, but at the company, is thinking about scaling for success and really thinking about global supply. So how many vials would you need if this is a success or even a partial success that one would want to move towards this manufacturing for a worldwide distribution for SMA. And there's multiple methods that this field, gene therapeutics field, are really moving towards suspension cells, baculovirus, as well as producer lines for the production process. I will end by just saying, and we heard this before by Dr. Reyna, it takes an army to really do the clinical developments as well as thinking about the basic and translational biology here. So I'm happy, I don't know if I'm taking questions or waiting till the end, but thank you so much for your attention. Great question. The short answer is that we had 3 to 4 pigs in that group that was weak and then delivered. We did CMAP Muni, and for the purpose of manuscript preparation and counting motor neurons and looking at motor neuron pathology, all of those pigs have been taken down for analysis. So we've not performed long-term studies in the non-human primates. We have plans to do so, to really investigate any respiratory distress, any other things that might be happening in long-term safety and efficacy. What I can say is in human clinical trials with AAV vectors, while no one has been in human trials with AAV9, there have been a number of human clinical trials with AAV vectors that transgene expression has been seen past 10 years. With no sense that it's wavering in its expression. Most of these are the hemophilia studies. There's some alpha-1 antitrypsin studies with intramuscular injections that shows long-term transgene expression. So our hope is that we won't have to go back in. There is potential to go back in, but it's a little bit more difficult than just going back and re-injecting without doing anything. And that is, once you've administered the virus, the body does develop a host immune response to develop neutralizing antibodies. So we've basically given the patient a vaccine against AAV9 because the body will develop neutralizing antibodies. There's a couple ways that you could get around that if you needed to go back in. One is go and use a new serotype that isn't neutralized. Okay, that's going through that whole process again on safety, et cetera. Other approaches, transient immune suppression and or plasmapheresis, which has been shown successful to actually transiently lower the neutralizing antibodies, go back in with a re-administration approach.
¿Otra pregunta? Brian, I'm going to ask you a question that many parents have asked me. How many patients do you have on your waiting list right now for the next treatment? So you have 3 enrolled.
There are 3 enrolled. I've heard, there has been no limit on the number of individuals calling. One of the issues with me being involved with the company is conflict of interest. So I'm not involved in any of the patient enrollment, other than knowing that there's a list of individuals who would like to get in on the clinical trial.
Right. So if now with the newborn screening, in progress at our institution, when we're finding these new babies born, the waiting period, it's quite hard for these families that are having a brand new baby, knowing now that there's possible therapy, even though it's not a therapy, it's still a trial. you have until 9 months of age. That comes up very quickly, right? So even though we may have some opportunities for these families, I hear this all the time when I'm doing newborn screening. It's a conflict, it's an ethical conflict for them, as it is for us, that we do have a trial but not really available. What would you say to those parents?
It's an excellent question, Sandra, and it's something that keeps me awake because we have people calling and who are days away and they're non-enrolling into the trial, and then they're past the 9 months of age, or someone who is a 10-month-old baby that would meet all of the criteria other than being under 9 months of age. And that's something that really is quite forceful and something that hurts clinicians as well as those sitting trying to run the clinical trial in the most expeditious, safe, and proper manner to be able to garner regulatory approval. So one of the things that we're working extremely hard on, so we're approved right now with FDA for 9 patients. We're trying to get the amendment in place for the CSF delivery route, which I hope I've impressed is as good as the systemic route of delivery, at least in the mouse, so that we can say that, okay, now the trial is open to 18 patients. So that might help the numbers, at least initially, but there's more than 18 patients. So I think this is where we've also come in with the help of the company and with funding from this company to run the clinical trial. Even though it's not a mountain of money, it's enough money to be able to start pulling in a little bit more experts in the design of the trial. So what do we really have to do this trial so that FDA might say, you guys have something? So we've actually brought in statisticians who have run many oncology trials, and they've basically suggested that we redesign this trial as this low, medium, high dose with then an expansion at whatever the safety dose is, in such a manner that one could create a pivotal clinical trial for regulatory approval in the most rapid manner. So I think these are sorts of things that we're trying to do, to be as quick to the pace as one can, but doing it judiciously for the proper ways. If Dr. Mendel makes a mistake on the clinical trial, even if this is, gene therapy is still on thinner ice, even though it's starting to thicken up. So we don't want to make a mistake and do something wrong in the trials.
And then just very briefly, why 9 months? Many parents ask that over and over again when they're still infants below 12 months of age. Why couldn't it have been 11 months and 29 days?
So I think in the initial design of the clinical trial with Dr. Mendel, John Kissel, many of the thought leaders, Kathy Swoboda, thought leaders in the field, basically there was the push to say, let me just say, it was a very strong fight with regulators, as well as back in the day, no one wanted to touch type 1 patients. And every trial should be designed for a type 2 or a type 3 patient. We disagreed with that. I think that the scientific and the clinical communities has now agreed that type 1 is an optimal patient population to get in. But now there's this concept of, get in early, especially for a biologic, a gene therapeutic, where, as many know, these babies can become extremely fragile, extremely quick, and the older they get, the higher the probabilities for that. So in the clinical trial design, it was put, initially we wanted, it was guided 0 to 6 months to try to limit patients from potentially going slightly down and getting worse on that cliff, because you don't want something bad going on. If your first patient dies in a gene therapy trial, I don't know, if your third patient dies in a short period of time in a gene therapy trial, in this field, we have to prove ourselves as being not guilty, as being the cause of death. And that's the main reason at this time to say go early. Dr. Mendel extended that window to 9 months to give, I think because some of the pushback, feedback from the communities to say, I have a 7-month-old, 8-month-old that is still strong and would be ideal into the trial.
I'd like to make a last question, probably. I was wondering whether this gene was also expressed or really infected other tissues. How sure are you it's completely directed to neurons or is not expressed in other tissues? It's really specific for neurons?
So no, it's not specific for motor neurons. It's in liver, it's in muscle, with a systemic route of delivery. Actually, with both routes of delivery, we can detect transgene in almost every single, or we can detect DNA and some transgene in almost every single organ. So both routes are actually systemic. CNS the CSF delivery enriches for CNS delivery, but it's still getting peripheral. Thank you very much.
Muchas gracias, Dr. Kaspar. Tenemos ahora nuestra última